So hello, my name is Jeanne Robinson, and as Peter said, I'm going to be chatting to you about invertebrates in the Cadzow Oaks and our study. So I should first acknowledge the support that we've had for this. The project wouldn't have been possible without the Clyde and Avon Valley Landscape Partnership, who gave us a large contribution towards our small funds, and the Glasgow Natural History Society gave us a small amount of money, and to SNH for actually encouraging the project and being supportive of access. So that's been great, and also the University of Glasgow for giving us the time to do it. A few of the things that we're going to go over, what inspired the project in the first place, a little bit about the site. You might know more about it than I do, but I'll tell you what I know, about the trapping regime that we've been using out there, the insects that we found so far, and through the trapping and the other things that we've come across, a bit about the kind of wider engagement that the project's encouraged, a bit about what next, and then hopefully there'll be time for questions if any of you have any. This very distinguished looking gentleman is Roy Albert Crowson. He lectured and looked after the museum collections at Glasgow University at the Hunterium from around 1949 until he died about 50 years later, so a massive tenure. And Roy Crowson was a monumental figure in beetle taxonomy. So he did loads of work collecting larvae, rearing things, and looking at the relatedness of things, but at higher level classification. And he wrote some of the definitive texts about identification and biology of beetles in the 1950s. So an incredible guy. He amassed quite a sizable collection. The local stuff remains with us at the Hunterium. The international material, the dissections and the slides all went to the Natural History Museum in London when he died. The reason he's relevant to our story is during the 1950s and 60s, from about 1955 to 1961, he did an awful lot of surveying around Hamilton High Park. He was interested in ancient oak woods in general, and he actually, in the archive at the Hunterium, we have his letters where he's pestering the Forces for Good to designate it as a triple SI because of the interesting invertebrates that are found there. So the other inspiration is personal. I used to live in Hamilton, and I used to go to the the small group of Cadzow Oaks. Strangely, whenever I go, the sun shines, and it's a strangely magical place, and proving probably that a love of old trees and oaks is genetic. That's my son, just aged 1, and probably expressing his joy at being near the Cadzow Oaks through the medium of dance. But when we all used to go, we would look through the fence, and you could see all the other trees, and it was like, well, these are lovely, but how do you get to look at those? So that was a big part of it. It was my nosiness and love of old trees. One of the principal things about it, it's a parkland oak site, and we'll talk more about that, which is important in its own right, but the most important thing probably is where it sits in relation to other woodland. We know about the importance of wildlife corridors. A lot of oak insects aren't very good at moving huge distances. Some are blind, some are flightless. The proximity of other aged trees at the right stage of decay and the continuity of that is one of the key factors. And of course it sits in, I think when I first wrote this talk, it was a candidate for a special area of conservation. Is it still just a candidate, or is it a special? Yeah. So it is now a special area of conservation. So it sits amidst a very important forestry area. And we know in terms of Scottish biodiversity that it really is very good. This is almost as good as it gets. Parkland oaks are really, really important for their invertebrate fauna. They're the most, in terms of insect species, they have the most insects associated with them of any plant. It's about 350 species that have been recorded in terms of oak, which is another reason why they're particularly interested to me, especially somebody who trains up students to look at diversity of invertebrates. One of the other, and in terms of their biodiversity, the thing that they're incredibly long-lived, they can reach incredible sizes, and when they become most magnificent in terms of diversity is when they start to get this rot at the core. Brown and white rot which are fungal led, will cause the entire centre of the tree to start to hollow out. And if you imagine that, what you actually end up is with a very unique microclimate. It's humid and it's sheltered, and even when it's raining in the site, some of the larger ones are quite nice to clamber into. We'll see a bit more of that. Also, the thing about the Parkland oaks is that they're spaced out, so they get sun on all sides. And that bell-shaped kind of classic oak canopy, you only get when they have enough space to produce that. Evidently, they'd just be trying to shoot up if they're in a dense woodland. What that does is it shelters out the lower branches. So the very big low boughs often eventually get shaded out and die actually on the tree. And then you get rot within those as well. And that's another really important habitat, because staying attached to the tree, it has a very specific environmental profile. So each of these trees really is like it's a mini ecosystem. There's an awful lot of good stuff going on there. This is quite a nice example. So you can see the front of the barks come away. So you can see that these, of course, the hollows in the trees aren't just used by insects. They're used by a vast number of other things. Anybody guess what these twigs have been deposited by? It is, yeah. Jackdaws. But my son's about 1.20 m tall, and you probably see it's over. It's at least twice the height of him, which gives you an idea of how many generations of jackdaws have probably reused that site. In invertebrate terms, this is quite important as well, because the one thing, it's lovely and sheltered in those hollows, but it's not very nutrient rich. And other things nesting in there offers valuable nutrient enrichment to the things that want to feed within that hollow. So, birds as insect food seems like a nice way round. Remnants of their feathers. This was just to give you some idea of scale of some of the hollows. You can get a full ladder and get a full 6-foot Canadian. So in addition to the kind of internal rot, the fungal communities that are associated with the trees have some really interesting invertebrates. And the ones that we would have liked to study, but we need a bit more time, are the mycorrhizal ones. So they have incredibly important fungal fauna around the roots as well. Chicken of the woods and the beefsteak fungus, two that have particularly interesting insects associated with them. You get a lot of tenebrionids and melandrids, beetles on chicken in the woods. And the beefsteak fungus, even though it's quite short-lived, is still important for flies like the heliomyzids. So there's some really good stuff that comes on the fungus. Fallen dead wood is another kind of vital resource that we were thinking about, because our studies were predominantly focusing on saprophytic species. We're looking at the beetles that Roy himself had collected back there. We've got a baseline study from the 1950s to compare to what we find in there now. His wife collected some spiders. We're also getting the spiders sent off for ID. And I'm working with Jeff Hancock, who was formerly curator of entomology, and he's really good on the Diptera. So those are our main focus groups, but we'll try to get a broader species listing. But the fallen wood, yeah, another vital resource. Most of it is just left. Veteran and established oaks support an incredible number of epiphytes, something like 300 species of lichen, many of them only found on veteran trees. And in European terms, we have some of the best epiphytic faunas on our oak trees going. You need clean air and things at the right stage of decay. And they're very important. So the lichens are important for things like the Scoptra and for moths, for both harbourage and for feeding. And the bryophytes support a similar. Groups. Oh, this was something else that I came across. I hadn't seen such a beautiful example. This was a slime mould. This is the fruiting body and the ethelium. What I love is the folklore. Apparently in Scandinavia, they call this thing, they call it the vomit of troll cats. It's also known as witch's butter. But a very mysterious structure that just appeared on the roots one day. So the oaks even support those. Wonderful biology. So actually, years slightly We started looking around in about 2016, ready to start in the spring of 2017. And from 2016 in the autumn, we were looking around and trying to think how we could best get a picture of what was happening entomologically in the site. There'd been lots of short-term studies before. Quite often, as funding became available, you'd get people out at the height of the season. So what we were keen to do was to get something that encompassed more of the year and targeted a broader range of things that wouldn't just be caught by hand netting. So a trapping regime seemed to be the way forward. So here we are inspecting the site. We had to think of things that would work with there being livestock on the site. That it's mostly sheep grazed at the moment, but in the summer months they do sometimes take in some cattle as well. So because they are just so curious. So the trapping regime that we spoke to somebody in the office, and they said it was that you have these wonderful enclosures that were for the young oaks that were planted in around the the kind of end of the 80s. 99 was that? Yeah, no, that's 1998, 99, wasn't it, that they went in? Yeah. And you said that the fences weren't required anymore, so we repurposed those and put emergence traps in. I don't know if you've seen these before. They're like Malaise traps, but they're sealed on all sides. So we loaded them with fallen wood and bits of loose bark and we sealed them up and we left them there to see what would emerge into the heads. We made these ourselves to fit into the enclosures, and also because at £80 a pop, we didn't really have a grant of the order that we could buy them. So we put 10 of these in all over the site. There was lots of fun time. The gentleman up the ladder was the one we saw climbing out of the rut hole. This is Justin Stevens. He's a fine artist from Canada who also has a great fondness for insects, and transpires his auntie used to be an illustrator in Hymenopteran parasitoid books. But he was very keen to help us make the traps. He's a fantastic seamstress or sewer, so he made the emergence traps. And he spent a lot of time experimenting with every bottle we could find for some fairly beautiful, largely impractical solutions. And we eventually... Oh yeah, here's the making of the emergence traps. I'll show you we also put in water traps. These you would most commonly seen for aerial trapping for things like flies, but we wanted something that we could put actually within the tree trunks because we knew this was where a lot of things were harboured. So they were made out of fizzy pop bottles where you just cut off the top and invert it. That makes a funnel, and you have the collecting fluid in the bottom. This was interesting. Trying to source the pop bottles, I realised how little ginger all of my friends drink. But we eventually amassed enough and spares to put in 10 of these around the site as well. So after all the bottle experiments, this is what we actually settled on. We got some second-hand bottles from one of the water cooler suppliers. They gave us their seconds. And we fashioned these that could be hung from the branches for further out in the canopy, and then which we called the aerial flight traps, and then aerial trunk traps, which we put up hard against the trunks to intercept insects in that area. These were the hardest to sight. These are rut hole traps. They're just tiny staples that are holding the net in place. But the idea was that we sealed up the rut holes, and there was a collecting head, and then anything that emerged from it within the period. The reason finding rut holes that weren't already occupied was a big part of the challenge. So we checked that they were clear before we sealed them up, because they're obviously very popular as bat roosts and as bird nests. And we've got a couple of very indignant chattering squirrels who were also not amused. So we eventually found 10. Some of the other things that we tried, we did qualitative sampling as well, going out with hand nets and looking under bark and those other things. We took samples of the heartwood of the rot, and we ran it in funnels. Another nice little experiment was with hand scourers. These have been used for collembola trapping before. The essence of it, I suppose, is a micro refugia. So sometimes what you don't need, you don't. Baited traps are good in terms of tempting things with food, but here it's just harbourage. It's just a nice place where they can feel safe, within a space, and you just take the scourers off after a period and then rinse them through or put them in the funnels and see what comes out. So putting up the traps was one thing. Then, of course, they had to be actively monitored. Most of them were ladder access. The hazard tape helped us locate them, and has come down now. We them. One thing about a year's trapping in the Scottish weather, oh, they took a battering. They really did. So we spent a lot of time. We've done quite a study of plumbers' sealants, actually, of which ones are best for keeping everything attached. We got through miles of gaffer tape and tried to hold things together in the interim to keep the sampling going, but it was quite a big maintenance regime. The other thing we discovered was we're getting the spiders identified, but we also did them a great service for the full year. On the right is our student Hamish. He's holding the custom spider brush, or a bottle brush, at which we'd have to clean the spiders out of the collecting heads, because you've produced this beautiful dry environment with a constant supply of food. They've probably never been happier. I mean, we did give them a sporting chance. They got flicked on the grass, and hopefully we intercepted a few things in between. Oh yeah, there's proof of some of the gaffer tape usage, a lot of string replacement. Sewing skills were also very helpful, because actually the nets started to perish towards the end. Some of them, it was a beautifully enclosed system for about the first 10 months, and then we had to start sewing them up to try and seal them up, just to make them last that bit longer, because remaking them wasn't really an option at that stage in proceedings. And the timber that had filled them, of course, had started to sink down, so we did just restock them. So this is the lovely volunteer Wendy sawing through dead wood, making it fit. So what did we trap? It's still relatively early stages in terms of the processing, so I'll tell you what we found so far. One of the first groups, which wasn't really one I'd expected to find in any great number, was in vast numbers in the emergence traps, and that is the thrips, or the thunderflies. Typically, you might think of them in association with flower heads or with cereal crops. So if you've ever worn a yellow T-shirt and been anywhere near a wheat crop at the wrong time, you'll know. But these are phleothripids, so this is a subgroup that feeds on fungal spores, and they are. And we've got an expert that works on them at the Hunterian, hence why we had someone who would actually look at them, which is a big thing, because they're quite tricky to ID and they're very variable. But we found a whole bunch of Hoplothrip species, including one that's new to Scotland. But it's predominantly southerly in distribution. I think there's probably our record from Durham and one from South Yorkshire, but it's definitely up here and doing very, very well. And we found those predominantly in the emergence traps, but also in the rot hole traps. So in the rot hole traps, so the rot holes that were sealed up, and the aerial trunk traps as well. And the Phytophthora expert was quite interested in this, because it's not a trapping methodology they'd typically seen for thrips. Another beautiful little one that's there in huge numbers is an ecophorid moth called the sulphur tubic, or Asperus sulphurella, one you're not really likely to mistake for anything else. Very distinctive and beautiful markings, but its caterpillar feed in decomposing wood, so that's why it was there in such vast numbers. And that's another one we found in the emergence traps in quite some number. The feather-horned craneflies are some of our biggest, most dramatic, most gorgeous, and they're typically associated with pretty established decaying wood and woodland sites. Really strong saprophylic link. This is probably one of the more common of them in Scottish terms, but there's still only a few sites that it's recorded from, Dicynidea bimaculata, and it's certainly alive and well at the Cadzow Oaks. We've had numerous specimens of this one. Really nice bright colours. Also sometimes referred to as the tiger craneflies, but there's other groups that are called that as well, so can be a bit. So this is not Well, to tell you the truth, so little's known about its biology. You'd probably call it it's definitely a decomposer, more strongly associated with leaf litter at the moment than anything else. But certainly, I've seen it crop up in a number of kind of saprozylic invertebrate studies. This is Cypher pulicaria. It's a nationally scarce species. What's interesting is Roy Crowson didn't find it. And if he didn't find it, you can almost argue it wasn't there because he's so good. But we found it in the emergence traps. And we've had some help from Mike Denson, who I think had done some surveys for you before, who's really into the rove beetles. So he's been identifying them as fast as we can throw them at him. And Charles Dickens style always says, can I have some more, please? We can't sort them fast. So that's quite nice. And he also came across this Quedius xanthopus, another kind of scarce rove beetle. But that one is found under dead bark, so a very strong saprozylic link. And we found one in the water trap as well as in the emergence traps. The ones on the right are the male and female of a little spider beetle called Tinus subpilosus, a nationally scarce species and a bit of a key kind of flag species for decaying oak. As a museum curator, it makes me cringe because usually when I see spider beetles, they're coming in to try and eat my collections. They are phenomenal detritivores. They hoover up all kinds of organic matter and they're great survivors. So learning to celebrate spider beetles was a good thing for me, overcoming my biases and prejudices, and of course having a lovely time out in the oak. And we found them in a whole variety of ways through the funnel extraction. So when we put them in the funnels with the rot material, in the rot holes and in the water trap. So they're very strongly affiliated with those tree hollows. The female's the voluptuous plump one on the right, and the male's the one with the lovely. So I realised I didn't sort the font out for this one because it was a later edition. But this is Thymalus limbatus. This is one of the species that you were asking about, Peter. So this is one that we have found so far. We've found a couple of specimens hand searching under bark in August. It's nationally scarce and there's very few Scottish records, so it's nice to know that that's still alive and well. And Crowson found it on numerous occasions between 1957 and 79, so we can confirm continued success and happiness. It's beautiful. It's like a little shiny golden jewel. Really nice one. We've been sending off the arachnids to Mike Davidson. So far, there's been nothing of great note. This money spider is an amberless species, but mainly because of its rate of decline rather than a kind of very limited distribution. But we've certainly found that, and we're on the lookout for some more. Those are what we're actively trapped for. One of the things about the site actually is the break in the age structure. So you've got the new trees that you've planted at the end of the 80s, you've got the old veterans, and there's just very little wet rot. So that's what's really missing. We couldn't find any damp rot holes. Usually when I walk around a site with Jeff, he's sniffing to walk towards all the things that everybody else would walk away from. But there was just none of it. There's very few hoverflies in general. There's not much moisture. The one place he did find it, and we heard him making happy noises, was around the stumps. So you'll see these are the sawn off oak stumps from when there was a thinning exercise. They're now at quite a nice state of decay. So it's particularly around the edges of the bark, where the bark's starting to come away. There's lovely little hidey holes for them, and some very rich fungal growth. And we found this lovely locally distributed nitidulid, Ceronia, that was just crawling across the moisture. Anybody recognise these? Yeah. I'd only seen them on David Attenborough, and I'd like to say it was my excellent kind of hunting skills that tracked them down, but they actually slapped me in the face when I came to track one of the traps in the rot holes. I was like, what's that? And I looked up, and yeah, there was a phenomenal mucus anchor. It was broad daylight, quite a hot day, which I wouldn't have thought were ideal mating conditions for something that needs a high level of moisture. But leopard slugs, they need things very moist. They do love dead timber. They also quite like basements and things. They don't need to mate to reproduce, but they do occasionally. If they do, it only tends to be once in a lifetime. And here, these are forming a phenomenal display. After their bodies have entwined, their genitalia entwine and form a phenomenal looking flower, and then the gamete exchange takes place. Something else to behold. And it takes so long, you just can't help but think how vulnerable they must be during that period. But yeah, they were lost in the moment, evidently. Yeah, and this is probably, I was expecting to be investigated by children's social services after this, actually, because I imagined Louis going back and telling people at school. This is a 4-spotted oak bug that I found squashed between the pages of my notebook. At that point, we hadn't sampled anywhere else, so it seemed to want to donate itself to science. So that's quite a good find. So there's about, oh God, how many species of gulls is it? Or is it about 60? 40. There's a big Wodger gulls that found on oak. It's one of the best species for galling species, and they're certainly looking very healthy at Cadzow. I love this leaf on the left, which you can see is just so thick with spangle and silk button galls. It's hard to believe the leaf can function at all. And nestled in it, you can play hunt the lacewing larvae. So there's one hanging about in there. And I really like this oak apple just because it seemed to have a smiley face. I thought perhaps I'd been out in the summer sun for too long, but see a nose and eyes. So it was, I mean, Jeff and I were the two kind of key members of continued staff. We recruited 3 curatorial assistants, as they were called, which were undergrads who helped us both in the field and in the labs. They got an awful lot out of it. We had 9 field volunteers from kind of other walks of life. So far, we've had over 1,200 hours, and it will be a lot more than that by the time the project's out. And I calculated we've got about 200 days of labour from professional ecologists and entomologists that have just helped us out along the way. We had 2 open days. I'll show you some pictures from those. And this is about the 5th incarnation of this talk. So there's been a lot of interest, which is lovely. And yeah, it will be over 120 people, I think, now. And it was very broad engagement. So yeah, people from 8 to 80. We've been quite far-reaching. Some pictures from one of the open days. The sun shone. It did, and there were many people. And the 8-year-old, his mum was trying to drag him off to rugby, and he did say, "Can I just no, not just now. There's a few more things I want to look at on the white sheet." So we did a bit of beating, and we had a look around, and we talked about the project. We had Scott Jones was a local photographer who came out and took some shots. I really love this one. It's a lacewing larvae. And if you don't know about them, they're just fantastic. I mean, they're only minuscule, so it's only those with good handling skills or microscopy that would probably appreciate them fully. Has a spiky back, and it uses it to impale the sucked out skins of its victims and other debris that it finds, which probably provides both olfactory and visual degrees of camouflage. And you see the jaws on it. There's no doubt it's a predator. Both the immatures and the adults feed on other insects. I love that shot. Yeah, so these were just some pictures that were also taken on the open day. So it was the open day and the butterflies were out. Yeah, the people were so happy. And we had this commas doing quite well at the site. And you might know, at one point there was some chit-chat about whether or not they were breeding here, but it was kind of established in 2010 that they're doing fine, and they're certainly doing quite well at Cadzow. The other reason I've taken this shot is it just gives you some idea to the level of agricultural enrichment that's going on around the base of the trees, which is probably a bit concerning. Certainly makes you wonder about the state of the mycorrhizal fungi with all of that going on. Let's enjoy the butterflies. So what next? So far, this is very much the tip of the ocean, and every time I walk into our laboratory, I'm reminded of this, because so 50 traps over the course of the year means there's a large bulk of material to process. So what you've seen are the start of what we will actually generate, because so far we have actually only identified about 1,000 individuals of about 120 species, and that's with the help of about 3 external experts, and it's probably only about less than a fifth of the material, but we're forging on. We've got somebody who's happy to look at the collembola. There's certainly an awful lot more beetles for us to sort through, and those will be the next target group. But things like the Psychoptera and the Parasitica we'll kind of probably keep and preserve, and then if anybody shows an interest, we'll have those samples that they can refer back to, because I think we'll probably have to draw the line somewhere. But there are some lovely. So I've got 4 more volunteers that are coming into the laboratory at the moment, helping me work through the material, helping do the provisional sorts so that we can partition it. I can look at the beetles, Jeff can do some of the other bits and pieces. Once you get them under the scope, we have found some lovely things. So I love pseudoscorpions. This is the first kind of set of sampling where I've really actually come across any. They're with Mike Davidson, and he's been amassing a few to identify them. So we'll hopefully have some IDs back on them. Britain's largest pseudoscorpion is found on oak. This might be it. But they're phenomenal little predators and really, really gorgeous. It's been nice for me. I've seen things that I hadn't seen before. Wonderful parasitic wasps. The one on the right-hand side, I was totally intrigued by. I was like, there, parasitic wasp with a handbag. How did I not know this existed in the British fauna? It's got a handle. What is that? What is that about? But it's actually the ovipositor sheath. And when it comes to lay its eggs in predominantly Homopteran prey, it does it over the top of its head, which must give it an element of surprise. It must be waiting for it to turn round, and it doesn't have to. So yeah, quite striking. And I just really like the antler-like antennae on the other eulophid parasite. Sod's Law. So the first, when we did the recce in the autumn of 2016, they had a phenomenal crop of beefsteak fungus. And a couple have dropped on the floor, took them back to rear. It got a bit past it, and I think a few things drowned. We got a couple of things out of it, but it didn't go very well. But we thought that's fine, because we've got next autumn nothing, hardly any at all. So it was a much drier year, and the fungal colonies weren't quite the same. Now that's quite disappointing in a sampling point of view, because quite a few of the key beetles were found in association with those. So if I ever get to the end of this, then I would probably like to go back and do a bit more work on the fungi. And the other thing that we haven't found yet is the cobweb beetle. So what's interesting about this is it's one of the few that you're more likely to encounter as a larva. Those very, very hairy little woolly bears sit on cobwebs, and they hoover up the spider's dinner. So you can see why they have to be quite as well armed as they are. So yeah, that's the cobweb beetle. So that one might be quite a nice one to do. We'd hope to do it as public engagement, and again, just time got away with it. We didn't. Yeah, we did the 2 open days, and then that was it done. Really, we'd run out of summer. But it would be. I think that could be quite a nice engagement exercise, getting people looking at spiders' webs. We checked them quite thoroughly, but no joy. We never found any on Hamish's brush. So that's largely it. We had also thought of doing some mycorrhizal trapping. We'd found some interesting insect traps that we thought might be quite good to do the root profiles, but again, it was just out of the scope and timescales. It was a total joy to be in the oaks for an entire year, from the luminescent green of the spring oak growth, which is absolutely day glow and beautiful. The winter, where you can really appreciate the sculptural charms of the veteran trees. So hopefully that's given you some insight into what we've been up to